Welcome to your Chapter 4 video lecture where we're going to discuss Section 4.3 on membrane transport. Now this section might be a little bit longer because I am going to play some animated videos for you. If you feel like you understand the concept, you can of course fast forward through that video portion, but I think it always helps to cement that information into your memory. So let's first talk about where membrane transport is going to take place. In your plasma membrane. So this will be a process of obtaining and eliminating substances across that plasma membrane, and we can classify it into two categories. We have our passive processes, which would include diffusion, and that breaks down into simple diffusion or facilitated diffusion. And facilitated diffusion can further be broken down into, is it channel mediated or carrier mediated? And the other type of passive process is osmosis. And our second category is active processes, in which we can have active transport, which can be primary or secondary. And if it's a secondary type of transport, it can either be symport, in which we are passing those two substances in the same direction, or anaport, in which we are moving the substances in opposite directions. And the other type of active process is vesicular transport. Is that through exocytosis, where we're releasing contents from the cell out to the external environment? Or is it endocytosis, in which we're bringing material into the cell in the form of a vesicle? And that has three different formats. We have receptor mediated, Pinocytosis, or phagocytosis. So we're going to talk about each of these processes coming up. First, let's differentiate between the difference between passive processes and active processes. Passive processes do not require energy, and that's ideal, right? And it's dependent on substances moving down their concentration gradient, meaning we're going from an area of a high concentration of, let's say, this ion of the brown circles, to an area of a lower concentration of these ions. And we have two different types, like we mentioned in the slide before, diffusion or osmosis. With active processes, it's going to require energy, and that's why we see ATP over here. We're going to see that it has the opposite of what we saw in the passive transport, meaning passive went from high concentration to low concentration. Notice how here we are going from an area of low concentration to high concentration. So we are going against the concentration gradient. And our membrane-bound vesicles have to be released in vesicular transport. So the first passive process we'll discuss is diffusion. This is when we have a net movement of a substance from an area of greater concentration, which would be this dye. I'm not sure exactly what they're representing here, but I think it's a dye, where we have greater concentration in the center there, and they are moving to an area of lesser concentration. The molecules and ions then are in constant motion due to kinetic energy. So almost think of these little molecules as moving around and bouncing off of one another. If it's unopposed, diffusion will continue until the substance reaches equilibrium and the molecules are nicely and evenly distributed throughout a given area. And here's a quick little video on diffusion.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Now let's talk about the rate of diffusion. It's going to depend on the steepness of the concentration gradient. The steeper the concentration gradient, the faster we will have a diffusion rate. So it's a measure of the differences in concentration within two areas. So here you could see we have a very high concentration of ions, whereas here we have a low concentration. So this is a steeper concentration. So you could think of it as a tall slide. Whereas here we have a higher concentration here, but it's not quite as high or much of a difference than what we have here to the right of our screen. So we have a slower rate of diffusion, so you can think of it as this lower slide. And temperature is also going to affect the rate of diffusion. It's going to reflect the kinetic energy or the random movement we have. The higher the movement of molecules we have with higher temperatures. So for instance, when you put a tea bag into hot water, you see that diffusion of the tea contents are going to diffuse throughout the water much more quickly or at a faster rate than we would if we put the tea bag into ice cold water. So simple diffusion is when we are having molecules pass between our phospholipid molecules. And you can see that we have no problem doing that with our small or nonpolar solutes. So we're including our respiratory gases like oxygen and also carbon dioxide. You can see here we have more carbon dioxide inside of the cell that we are trying to move out of the cell. And also some fatty acids and ethanol and urea are able to do this. These movements are not regulated by the plasma membrane, but just are taking place depending on the concentration gradient, and will continue to move as long as that gradient exists. Now let's talk about facilitated diffusion. This is a transport process for small charged and polar solutes that aren't going to be able to move across as easily as what we just saw. And we have two different types. We have our cell-mediated diffusion, facilitated diffusion, and carrier-mediated facilitated diffusion. Our cell-mediated diffusion is when we have movement of small ions through water-filled protein channels. And you could see an example of this pictured here to the right. Our leakage channels are going to be continuously open. So we would be able to move from an area of high concentration to low concentration, no problem. Other times we will have gated channels, such as this voltage-gated sodium channel. These are typically closed, and the only time that they will open is in response to a stimulus for a fraction of a second. So with the voltage-gated sodium channels, our cell needs to reach a certain voltage before the channel is going to open. So once that voltage is opened up, then we're going to have the ions, the sodium ions, be able to move through from a high to low concentration. And like I mentioned, this only happens for a fraction of a second, and then they're going to enter an inactivated phase in which we aren't going to be able to open it in response to a new signal. And this is typically referred to as a refractory period. And it's a very important type of normal functioning that we have in muscle and nerve cells. So more on this when we get into those systems. Now let's talk about carrier-mediated diffusion. This is when we have small polar molecules that need assistance moving across the plasma membrane, and it does that through a carrier protein. We have the binding of our substance, in this case it is glucose, over to our carrier protein. and we are going to release the substance on the other side of the membrane. We're still moving the substance down their concentration gradient, meaning we have a high concentration out here and a low concentration over here, and we are using the uniporter here, meaning it's a carrier transporting only one substance, and the number of channels and carriers determines the max rate of the substance that we are transporting. Let's take a quick 1-minute look at this video on facilitated diffusion.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
And now for our second passive process, that is going to be osmosis. This is when we're talking about movement of water, not the solutes or ions, which would be the solutes. So we're going to have a passive movement of water through selectively permeable membrane. So our membrane is going to allow for the passage of water, and these red molecules with the blue representing the hydrogen there are water molecules. So you could see that they are easily passing through the plasma membrane. And our membrane prevents passage of most of the solutes. So you could see some of our solutes like glucose out here, protein. I'm not sure what's represented by the blue dot here, but basically what they're trying to depict is that these solutes are being kept out unless they have a specific protein that is going to help move them or transport them across. And the differences in water concentration on either side are going to be based on what they're attracted to. Water is always attracted to solutes, so wherever it has the more solutes is where it's going to want to go. So here we have two ways in which water is crossing the membrane. It can slip between the molecules of our phospholipid bilayer, or it can move through these special channels known as aquaporins. These are integral protein water channels. And we have two types of solutes. We have our permeable solutes that are able to pass through the bilayer, and an example of this would be like oxygen or carbon dioxide or urea, which are not pictured in the figure to the right. And our non-permeable solutes are going to be prevented from passing through the phospholipid bilayer. So these would be like our charged molecules, such as sodium or chloride, or even our large solutes such as glucose, or even our proteins, which are much larger. And our concentration gradients across the plasma membrane are going to be between the cytosol and the interstitial fluid. So what I'm talking about there, if I flip back to this image, the cytosol would be here inside of my cell, and the interstitial fluid would be here or outside of my cell. So that's what we mean by that. And the solutes are prevented from moving across the bilayer. So we're going to imagine that this tube here is like the cytosol and the interstitial fluid. And here, the semi-permeable membrane is like our plasma membrane. So the concentration gradient is going to cause our water concentrations to exist within these two different spaces. The greater concentration of solutes contains a lower concentration of water. So notice this is our situation before osmosis takes place. Then when osmosis, or the movement of water, takes place, we are going to see that water is going to be attracted to those increased number of solutes. So now water has shifted from side B over to side A, and water is basically moving down its concentration gradient until an equilibrium is reached, meaning in the amount of water that we have, those solutes are kind of evenly distributed. And when that takes place, we get this osmotic pressure that gets created. This is the pressure exerted by that movement of water across the semipermeable membrane, and it's due to the difference in solution concentration. The steeper the gradient, the more water that's going to be moved across by osmosis, and we have greater osmotic pressure. We also have hydrostatic pressure, and this is the pressure exerted by a fluid on the inside wall of its container. So we use this term a lot when it comes to discussing blood vessels and the pressure of blood, the fluid, on the inside of the blood vessel wall. Now, before we talk about osmosis and tonicity, I want to show you a little video on osmosis.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
So this video described a little bit about tonicity. Cells can gain or lose water through this process of osmosis based on the change in the cell's volume and osmotic pressure. So when we use that term tonicity, it's referring to the ability of a solution to change the volume or pressure of a cell by osmosis. And when we have isotonic concentration, that means we have equal concentrations of water in and out of the cell. And you could see this illustrated in this figure over here of our red blood cell, of the water components moving in and out, so that everything is in equilibrium, and therefore we will eventually have no net movement of water. Examples of this would be normal saline with a concentration of 0.9% sodium chloride, which is commonly used in IV solutions. And our hypotonic solution is when our solution has a lower concentration of solutes, and instead we have a higher concentration of water than in the cytosol. So then what happens is water is going to enter the cell. So our erythrocytes, or red blood cells, would have water enter the cell if we were to place it within pure water. Water moves down its concentration gradient and into the cell. So we see that we get an increase in volume and pressure of the cell, and what can result is lysis or rupturing of those red blood cells, which would obviously kill those red blood cells, and that is referred to as hemolysis. When we have a hypertonic solution, that means we have a solution with a higher concentration of solute than our cytosol within our cell. So this would be like placing our red blood cells in 3% sodium chloride water, meaning we have very salty water. And so water is going to move down its concentration gradient, and it will want to move from inside of the cell out. And that'll decrease the volume and pressure of the cell, which will cause it to shrivel or shrink. And this cell shrinking is called crenation. And you could see that pictured over here in this electron microscope micrograph. So if you remember back to the beginning of this section, we discussed how our active processes are organized into active transport and vesicular transport. Our active transport is going to allow movement of a solute against its concentration gradient. So we're going to go from an area of low concentration into an area of high concentration. And we're going to maintain this gradient between the cell and interstitial fluid. And our source of energy will determine whether this movement is primary or secondary. So let's see exactly what am I talking about. First, let's discuss primary active transport. We are going to use energy here by directly breaking down ATP. Remember, we go through the process of phosphorylation, in which a phosphate group added to a transport protein will allow changes to the protein shape and movement of the substance across the membrane. So let me go through this again. We have a solute that is going to bind to the transport protein. ATP comes along. We break off one of those phosphate molecules through the process of phosphorylation. So now our molecule left over is ADP because we only have 2 phosphates left. And that inorganic phosphate attaches to the protein, and we get the conformational shape change here of our protein that will cause a release of our solute into this extracellular space. And then that protein will change its conformation again, and the phosphate can then detach. So this happens in ion pumps. Our cellular protein pumps are going to move ions across the membrane. It helps to maintain an internal concentration of the ions. So an example of this would be calcium pumps. We have our calcium that would bind to our protein here, and we could find these in our red blood cells. And it really helps to prevent cell rigidity from accumulated calcium, and our red blood cells are going to remain flexible enough to move. So you can see that the calcium is being pumped into the interstitial fluid outside of our red blood cell. Another very important active process or active transport is using our sodium-potassium pump. This is going to be an exchange of ions, and we're going to move one type of ion into the cell against its gradient while moving another type of ion out of the cell against its gradient. Essentially what we are doing is moving 3 sodium ions out of the cell, and we are moving 2 potassium ions into the cell. And our plasma membrane preserves the steep gradient differences. So let's take a look at an animation of what this process is like.
The sodium-potassium exchange pump is an example of an active transport process. Active transport processes can move substances against a concentration gradient, and they require energy expenditure in the form of the breakdown of ATP. The sodium-potassium exchange pump moves sodium ions out of the cell and potassium ions into the cell. The carrier protein for sodium-potassium exchange is embedded in the cell membrane. 3 sodium ions and an ATP molecule can bind to the carrier protein on the inside of the cell membrane. The ATP is broken down to ADP and phosphate. Simultaneously, the carrier protein changes shape and the sodium ions are transported across the membrane and released on the outside. In its new conformation, the carrier protein can now bind potassium ions on the outside of the cell. 2 potassium ions move into the carrier protein from the outside of the cell membrane, and the phosphate attached to the carrier protein is released. The carrier protein changes shape again, reverting to its original conformation, and delivering the potassium ions across the membrane to the inside of the cell. It is now ready to repeat the process of sodium-potassium exchange.
And of course, I have to add this little Drake video of where we're placing the sodium and potassium ions. So again, cytosol is going to be inside our cell and extracellular is outside our cell. So you can see that we are placing the potassium inside our cell and the sodium outside of our cell. And of course, ATP is present to drive this entire process. So now let's talk about the secondary active transport. This is when we're moving a substance against the concentration gradient once again, and we have to use energy from movement of our second substance down its concentration gradient. So kinetic energy here is going to provide the power to pump our other substance. In this case, we have our sodium-potassium pump, but you could see in our secondary active transport, we are going to move across one sodium ion along with a glucose ion. So this is our symporter because we have 2 substances moving in the same direction, whereas our sodium-potassium pump would serve as our antiporter. So our previous slide was an example of how the primary active transport and secondary active transport work together. And within our secondary active transport, it doesn't necessarily have to always be a symporter. Our secondary active transport can also be an antiporter in which those 2 substances are moved in opposite directions. Now let's talk about vesicular transport, which is also known as bulk transport. And remember, that was subdivided into exocytosis, where we move substances out of the cell, and endocytosis, in which we move substances into the cell. Both of these processes are going to involve energy input to transport large substances across the plasma membrane by a vesicle. What we mean by vesicle is that it is a membrane-bound sac filled with materials on the inside. So let's look at exocytosis first. This is when we have large substances that are secreted from the cell, and our macromolecules are going to be too large to move across the membrane on their own. So we put them into the secretory vesicle, and then we'll have that vesicle kind of bud up to that plasma membrane, and we will get a fusion of that vesicle with our plasma membrane. We need ATP in order for this process to take place, and the contents inside that vesicle will then be released to the outside of the cell following that fusion that took place. An example of this would be when we release neurotransmitters from our nerve cells. Now let's take a look at endocytosis, where we're moving contents inside our cell. We are going to take up a large substance from our external environment, meaning outside of the cell, and our steps are similar to exocytosis, but they're kind of in reverse. So what we're going to do is move the substances toward the plasma membrane here, and then we form a vesicle from the plasma membrane. And then through that vesicle, those contents can travel over to where they need to go for cellular processes to take place. And we have 3 different types of endocytosis. We have phagocytosis, which is pictured to the right here. Phago means to eat, and cyto is referring to cell. So this is like our cell eating. And what we're doing is we are going to take a particle from outside of the cell, and form these large extensions called pseudopodia. Pseudopodia. Pseudo means false or fake, and pod means foot. So they're like these foot processes that are causing an invagination and capturing that particle. And then they're going to kind of pinch off and surround that particle to enclose it in a membrane sac. This sac is then internalized, and the contents would be digested after fusing with another cellular structure called a lysosome that contains digestive enzymes. And only a few cell types are able to perform this, such as white blood cells, like our neutrophils and macrophages, if you've heard of those types of cells, and will digest microbes as well. Our other type of endocytosis is pinocytosis. So I want you to think as phagocytosis as almost taking in more solid, like think food type of things. And pinocytosis is more smaller particles or fluid. So this is why pinocytosis is also called cell drinking. We're going to internalize droplets of interstitial fluid that contain dissolved solutes. So we'll have multiple small vesicles that are formed, and this is performed by most cells. And the last type is through receptor-mediated endocytosis. We are going to use receptors here. So I know it's kind of hard to see, but this yellow Y-shaped structure is a ligand receptor, and the blue structure is the ligand. So you can think of the ligand again as like a hormone or a neurotransmitter. So when we have that in place, what starts to happen is we get this invagination, and almost like the plasma membrane is pinching off here. So we notice that this starts to, or the receptors and ligand complex rather, starts to accumulate at these special membrane regions that contain these special proteins called clathrin proteins. And they are found on the internal surface of the membrane here. And the clathrin, if I can pronounce it right, coated regions of the plasma membrane here are the ones that are housing this ligand receptor complex. So it starts to fold inward, and we call this a clathrin coated pit. And it starts to deepen and pinches off, and eventually we get this clathrin-coated vesicle. This is then going to move into the cytosol, and this clathrin-coated portion has to be enzymatically removed before the vesicle can move into intracellular destination, wherever it's going to go. After the clathrin is removed, then the ligands can be detached from the receptors and can be stored, modified, or destroyed. And these receptors just travel back up to the plasma membrane and sit right up here waiting for another ligand to come and attach. Now, an example of this would be like the transport of cholesterol from blood to the cell. Our cholesterol is bound to our low density lipoproteins, or LDL, and they move from the blood into the interstitial fluid and then bind to LDL receptors that they have in the plasma membrane, and the LDLs would then be internalized. And I put this figure in here. This is a concept overview to discuss the passive processes that we mentioned and active processes. So I highly recommend checking this figure out to review all of these processes. I'm going to play a video for you showing you exocytosis and endocytosis.
Endocytosis is a process in which cells take in materials that are too large to pass through the plasma membrane. In this process, the plasma membrane surrounds and engulfs large particles. Cells use three basic types of endocytosis, depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell, or an organic fragment, the process is called phagocytosis. If the material is small enough to be suspended in the extracellular fluid, such as certain proteins, the process is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is a process in which materials are discharged from a cell. In exocytosis, secretory vesicles migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
So that is it for this section. If you would like to review any of these videos, they are available in APR for you to see. And if you have any questions, please contact me.